Welcome to our video lecture on topic B1.1 Lipids. Our guiding questions for today: in what ways do variations in form allow diversity of function in lipids? And then we're going to also talk about how carbs and lipids compare as energy storage compounds, which is our first objective, comparing and contrasting that energy content of carbs and lipids. We're going to discuss examples of lipids. We're going to look at fatty acids, including lots of variations in the forms of fatty acids: saturated, unsaturated, cis and trans unsaturated, and then polyunsaturated. We're going to look at condensation reactions that produce triglycerides. We're going to state the functions of lipids. We're going to review the structure of phospholipids. We talked about these quite a bit when we looked at membranes. We're going to describe the structure and function of steroids and wrap up with a look at why high cholesterol in the bloodstream is not good for us. And here's a lovely tea table helping us compare and contrast carbs and lipids. BioNinja with the amazing mnemonic device to help us remember different ways to compare and contrast. Carbs like glycogen, or this guy right here, sucrose. Our lipids, triglycerides, or here is a fatty acid. In terms of storage, how much energy they store. Our carbs are a little bit shorter term energy storage. Our lipids are longer term. In terms of osmolality, in terms of how these guys affect how much water moves into and out of the cells, carbohydrates have a larger effect than do the lipids. So we can put lots of lipids into a cell and not worry about water following. Digestion, those carbs are easier for us to digest. We can pull that glucose out of the glycogen and get it into aerobic and anaerobic processes to make some ATP pretty quickly. Our lipids takes a little bit longer, and we can only use them in aerobic respiration. How much ATP can I get out of each? Way more out of lipids than out of our carbs. There's still a lot of energy in carbs, but not quite as much as in lipids. This is kilojoules per 100 grams. Sometimes we also talk about calories per gram. So for our carbohydrates, it's 4 calories per gram, whereas in our lipids, we've got 9 calories per gram. So again, quite a bit more energy stored in our lipids than in our carbs. But then in terms of solubility, carbohydrates for the win. Carbohydrates are quite a bit more soluble when they're in their smaller forms, so they're monomers and dimers, sucrose and glucose, super easy to transport around the blood. Our lipids are hydrophobic, and so they're much more difficult for us to transport. Here we have lots of examples of lipids. Oils, like this guy, are going to have pretty low melting points. They're liquid at room temperature. Notice that the fatty acid chains, we'll talk about this on the next slide, they have these little bends in them. That's not present in the fatty acid chains of this fat in butter versus oil. These fats do have higher melting points than do our oils. They're solids at room temperature. They start to melt at about body temperature, so getting close to 37 degrees Celsius, they'll start to melt. Waxes have even higher melting points. They will stay solid at room temperature, and they also stay solid at body temperature. So their melting points are going to be even higher than 37 Celsius. And last up, we're going to look at steroids. Steroids, like cholesterol, are going to have this 4-ring structure, and then most of them have some kind of tail as well. We're going to spend lots of time here looking at the structure of some fatty acids. Fatty acids can be saturated. That means they don't have any double bonds between the carbon chains. All of our fatty acids do have on one end a carboxyl group. The carboxyl group is a carbon double bonded to an oxygen and also single bonded to an oxygen, and then there's a hydrogen at the end. That's our carboxyl group. This is actually what makes the fatty acid an acid. Attached to that carboxyl group is this crazy long chain of carbons and hydrogens. We have lots of carbons, hydrogens, carbons, hydrogens, carbons, hydrogens, all along this crazy chain. If I have no double bonds between the carbons in the chain, this double bond here makes the carboxylic acid an acid. That guy has to stay. But if I don't have any double bonds between the carbons in the chain, then we call it saturated. These saturated fats tend to be more in us animals. It helps to keep our fats at close to solid, which helps us to maintain some constant body temperatures. It is a lovely, lovely insulator. Unsaturated fatty acids are now going to have some double bonds between the carbons in that long chain. So I've got a carbon here, I've got a carbon here. Notice we now have a double bond. Let's review the chemistry of carbon super fast. Remember that carbon has a valence of 4, which means that it needs to make 4 bonds. In our fatty acid, each carbon is usually bonded to 2 carbons and then 2 hydrogens. If I add a double bond between some of those carbons, this guy now has 1, 2, 3, 4, 5 bonds. That's too many. It can only have 4. And so we delete one of those hydrogens. So now we have unsaturated it because it has fewer than the max possible number of hydrogens. If we take those hydrogens away on opposite sides, oh, that's a hydrogen, not an oxygen, sorry. I was circling and now it looks like an oxygen, it's a hydrogen. So carbon to hydrogen, if I take the other hydrogen away on the opposite side, there used to be a hydrogen here, there used to be a hydrogen here. I've taken them away on opposite sides. We call that a trans unsaturated fatty acid, trans because it's across. This kind of helps to make that fatty acid stay straight-ish. And so these guys have properties that are similar to the saturated fatty acids. They tend to have higher melting points. They tend to be less healthy for us. And we'll talk about that a little bit more on the next couple slides. We have another kind of unsaturated fatty acid called cis-unsaturated fatty acids. We're still throwing in that double bond between our 2 carbons, but now we're leaving hydrogens on the same side, cis for same. And then I have this hole of where the hydrogens used to be on the same side. These molecules are like, I'm going to take advantage of all that empty space and bend. These hydrogens are going to repel each other. They're going to push away. Because they have lots of space to push away now, they do. And so our unsaturated fatty acids that are cis, holes on the same side, hydrogens on the same side, are going to end up with a little bit of a bent configuration. This makes them healthier for us. We can actually make multiple double bonds along our fatty acid chains. Notice this guy has 1, 2, 3. Notice that all of them are cis. So I've kept my hydrogens on the same side. There's a hole here. Hydrogens on the same side. There's a hole here. Hydrogens on the same side. There's a hole here. So this is a unsaturated fatty acid, poly because it has many double bonds, and it is all cis, and so it's going to be super, super healthy. Sometimes we shortcut, and so you might see shapes like this. I don't love this as much because even though we can see that there are multiple double bonds, we cannot see if they're cis or trans when it's in this shape. This shape is better because I can definitely see that they're all cis. This is called an omega-3 fatty acid. You might have heard of that if you're looking at the vitamin aisle at the grocery store. So it's called omega-3 because if we count 1, 2, 3 carbons from the end, we have that double bond. The first double bond comes at carbon number 3 from the carbon chain end, and that's going to make this omega-3. If I didn't have a double bond here, if it was a single bond here, and I didn't have my first one until 4, 5, 6, that would make this guy an omega-6 fatty acid. So that's where those come from when we're looking at our diet supplements. Another dietary supplement that we sometimes see are medium chain triglycerides, MCT oils. These are also healthy for us. Medium chain because literally like it's a medium chain, not a crazy long chain of fatty acids on that triglyceride. We looked on the previous slide at that MCT, that medium chain triglyceride. Let's talk about what even are triglycerides. So you probably guessed from the tri part that there's 3 somethings in there. And the 3 are 3 fatty acids. The 3 fatty acids are attached to this backbone that we call glycerol. So this is one glycerol molecule. And then we have 1, 2, 3 fatty acid chains attached to it. This makes it a triglyceride. The function of our triglycerides, they are super good for energy storage. We have, oh my goodness, so much energy stored in all of these chemical bonds. So much energy stored in all those bonds. We use these triglycerides also to protect organs. So around the kidney here, you can see lots of fat. That fat outside the kidney is actually good because it's protecting the kidney from whatever is trying to run into it inside the body. If there was fat inside the kidney, that would be less good. Triglycerides are also used for insulation, so Arctic mammals, other organisms that live in the cold water have oodles and oodles of blubber. That blubber is lots and lots of triglycerides. It helps to insulate the body of these animals from that cold water. It's a good protectant. It also helps to increase the buoyancy. So these guys are better floaters because they have all that fat in there. How do we make triglycerides? I wonder if you are wondering. Let's talk about it. So again, here we have our glycerol molecule. Glycerol molecules have 1, 2, 3 carbons, and then 1, 2, 3 OH groups. Those groups are called hydroxyl groups. They've got 3 hydroxyl groups. Here are our fatty acids. I have a fatty acid here. Here's that carboxyl group, similar to a hydroxyl because it's got that OH piece, but it also has a double bonded O, which makes it carboxyl instead of just hydroxyl. This N is telling us that I have some large number of C's and H's. So I could have C, 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 C with all those H's, 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 H's repeating for some long number. And then at the very end, we have the CH3, 1 carbon with 1, 2, 3 hydrogens at the end. When we build our triglycerides, we're going to take the H from the glycerols, hydroxyl. We're going to take the OH from the carboxyl group of the fatty acid, and you guessed it, 2 H's and an O makes some water. So we're going to pull out a water that makes this a condensation reaction. Condensation reactions are anabolic. I'm definitely taking some, let's say medium size, not so small these guys, some medium sized molecules, building them together to make a giant molecule. We're going to do that along every single hydroxyl group of the glycerol molecule. So I took off 1 here. I'm going to take this H and this OH. We're going to make another water. This H and this OH. We're going to make a third water. And then we're going to make some bonds. This O that's left and this C that's left, they need some bonds. And so they will find each other. This O and this C will find each other. This O and this C will find each other. And then we end up with these bonds here, 1, 2, 3. And these are called ester bonds. And they make our triglycerides. So I now have that glycerol with 1, 2, 3 fatty acid chains, tri for triglyceride. Sometimes we do not put 3 fatty acid chains onto our glycerol molecule. I might put only 2. And so I'm going to keep here my glycerol backbone. Here's our glycerol backbone, that 1, 2, 3 carbons with an OH. That H got taken away when it made water with the piece of the carboxyl group that we took off of the fatty acid. And so here's glycerol with fatty acid. Here's that second carbon in the glycerol with the fatty acid. That third carbon in the glycerol, instead of sticking it onto a fatty acid, we're going to put it onto a phosphate group. We now have a phospholipid, and we remember from our unit on membranes that we have a polar phosphate head and those 2 fatty acid chains. What holds those 3 guys together is glycerol, just like in our triglyceride. Remember that lipids are hydrophobic. This means that they are not attracted to water. They don't dissolve well in water, but they will dissolve in nonpolar solvents like our acetones. They tend to be more attracted to other nonpolar substances, which is why these guys assort themselves toward each other. They are attracted to each other. They're not attracted to the water. Phospholipids are super cool because they are both hydrophobic and hydrophilic, which makes them amphipathic. Not all of our lipids are amphipathic. This is a special thing, a special property of just our phospholipids. We're going to look also at these steroids. Steroids are one last class of lipids that we're going to look at. Steroids have these 4 fused rings, 1, 2, 3, 4 fused rings, and then most of our steroids also have some kind of tail. This guy is cholesterol. A lot of our reproductive hormones are based on cholesterol. And so testosterone looks a lot like cholesterol. Here's estrogen, which is sometimes also called estradiol, looks again a whole lot like cholesterol. What's kind of great about these steroid hormones, these lipid hormones, is because they are nonpolar, they can actually pass right through the cell membrane. They're a little big, so they don't go crazy fast, but this is simple diffusion. We do not need any protein pumps or channels to help make this happen. Testosterone, estrogen, also progesterone very easily can pass through the membrane because they are nonpolar. Remember that our cholesterol will sit in the membrane. It'll sit in the membrane here because also this chunk of it is nonpolar, so that part's going to sit between the fatty acid chains. This one tiny little hydroxyl group is a little bit polar, and so it's the piece that sits between those phosphate heads. One other look at a steroid that you might have heard of before is cortisol. Cortisol is one of our stress hormones. So another one of these 1, 2, 3, 4 fused rings with some kind of tail that we use as a hormone. So that cholesterol in our cell membranes that helps to maintain membrane fluidity, that's good. But sometimes our cholesterol and other fats can build up inside our blood vessels as plaques like this. So this is just lots of gunk, lots of fatty gunk that has stuck to the inside of our blood vessels. It's reducing the space that our blood has to be able to travel through the blood vessels. This is known as atherosclerosis, or hardening of the arteries. And it's bad because if I can't get my blood flowing, we're going to have a pressure build up back here. That's a really bad color to use. Pressure build up, which is going to lead to high blood pressure. That makes our heart have to work harder. Our heart shouldn't have to work harder. It works hard enough already. So these are problems. It's all related to these lipoprotein complexes that we have floating around in our bloodstream. So remember from a previous lecture when we talked about water and how water is such a good transport medium, except for things that are non-polar because they don't dissolve very well in water. So we have these lipoprotein complexes. And a lipoprotein complex is going to be this bubble of a phospholipid monolayer. So we have our phosphate heads here, which will very happily associate with the water of the plasma in our blood. But we have only a monolayer, so those tails point in, which makes the whole inside nonpolar. And here we have our cholesterols and our triglycerides, and then we can transport this whole bubble of fat through our bloodstream because it's wrapped up in this phosphate head coating. We have different kinds of lipoprotein complexes. This is very low density lipoprotein, low density lipoprotein. We can also have some high density not that high, this high density lipoproteins, and these are good because they move through the bloodstream pretty well. They do not stick too much. But if we have more and more of these lower density lipoproteins, these guys are fluffy and sticky, and they start to stick to the insides of our blood vessels. Some stickiness is normal. Too much stickiness, so this high cholesterol level, notice how much space that cholesterol is taking. There's very little space left for the blood to be able to flow. There's very little space left for that blood to be able to flow through the vessel. This is not good for us. So watch your cholesterol levels when you have your annual checkups. Make sure that your doctor is talking about how your cholesterol is looking. Try to eat more of those cis fatty acids, more oils, fewer fats. Stay away from margarine because even though it sounds like it should be good for us, it's not. Reduce your butter intake, more oil, less butter, no margarine. And on that health note, my friends, we have accomplished our lecture on lipids. We talked about how variations in form allow for the diversity of function in lipids. We talked about triglycerides and cholesterol and phospholipids. We also looked at carbs and lipids as energy storage compounds. Carbs are more easily digested and are more easily transported, but lipids can store more energy, which is exactly objective number 1. We talked about different examples of lipids, those oils, fats, the waxes, the steroids like cholesterol. We looked at the structures of fatty acids. We had saturated and unsaturated, cis and trans, and then polyunsaturated. Cis is healthy. Eat more cis. We talked about those condensation reactions that produce the triglycerides. We looked at some of the functions of lipids. We talked about phospholipids super briefly. We already know so much about those guys. We talked about the structure and function of steroids. Remember that they have those 4 fused rings and then some kind of tail. Steroids are like cholesterol and cortisol and progesterone, estrogen, and testosterone. And then we talked about how high cholesterol in the bloodstream can lead to some pretty big health problems. And so eat less butter and definitely eat no margarine. Good work today, my friends.